Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Selman, Waksman and Albert Schatz report, published in 1952 and entitled Actinomycin D, a new antibiotic active against certain bacteria, emerges as a pivotal contribution, deeply embedded within the burgeoning field of antibiotic research. This investigation will undertake a section-by-section analysis of this paper, rigorously evaluating its claims, methodology, and implications concerning the novel antibiotic, Actinomycin D. A comprehensive examination will illuminate the core scientific arguments, the evidence presented to support these assertions, and the overall impact of this work on the trajectory of antibiotic development. Section 1, Introduction and Overview of Antibiotic Research. The introductory section of the Waksman and Schatz paper sets the stage by positioning actinomycin D within the prevailing context of antibiotic discovery. Before diving into the specifics of their findings, the authors implicitly acknowledge the rapid progress made in the field of antibiotic research, particularly following the discovery of penicillin. Early claims center around the potential of the Actinomyces genus as a fertile ground for the identification of novel antimicrobial agents. The central assertion revolves around the premise that continued exploration of soil microorganisms, particularly Actinomyces, would yield further compounds capable of inhibiting bacterial growth. In evaluating this foundational claim, one must recognize the inherent inductive logic. The success of previous antibiotic discoveries, rooted in the screening of microbial natural products, forms the basis for this prediction. While this approach possesses inherent merit given past successes, it also carries the risk of overlooking alternative sources or mechanisms of antimicrobial action. The authors further assert the significance of identifying antibiotics active against bacteria resistant to previously known agents, alluding to an already developing problem of antibiotic resistance, a prescient observation for its time. This call for novel antibiotics constitutes a crucial argument, justified by the evolving landscape of microbial adaptation. The initial focus on actinomyces becomes strategically meaningful. The earlier work on streptomycin, an antibiotic also derived from actinomyces, lends credence to this specific research direction. This strategic element highlights the importance of building upon previous successes when charting new scientific territory. Section 2, Production and Isolation of Actinomycin D. The second section details the production and isolation of actinomycin D. Claims here concentrate on the specific fermentation process utilized to cultivate the Actinomyces strain responsible for producing the antibiotic. Waksman and Schatz describe the use of aerated submerged cultures, a methodology designed to optimize microbial growth and metabolite production. Their initial claim asserts that this specific fermentation technique is conducive to the efficient production of actinomycin D. Critically evaluating this, one notes that while submerged culture is a common method, its effectiveness depends largely on the specific nutritional requirements and physiological characteristics of the producing microorganism. The author's present data, albeit limited, suggesting that manipulating the nutrient medium results in variations in antibiotic yield. This variability implicitly establishes the importance of optimizing fermentation conditions for maximum production. They subsequently describe the isolation procedure, relying on solvent extraction and chromatographic techniques to purify the antibiotic from the fermentation broth. This sequence of steps, involving sequential extraction with organic solvents, attempts to achieve selectivity in the separation process. The success of this isolation procedure hinges on the differential solubility of actinomycin D compared to other components present in the fermentation broth. A claim about the purity of the isolated compound is implicitly made. The data presented in the paper lacks comprehensive characterization of the isolated product. While they do describe the use of chromatographic separation, a more thorough analysis, including spectroscopic methods, would have bolstered the assertion regarding the compound's purity. The reliance on bioactivity assays as the primary means of detection and quantification presents inherent limitations when determining the absolute purity of the isolated material. Section 3. Antibacterial spectrum of actinomycin. This section presents the claims surrounding the antibacterial spectrum of actinomycin D, detailing its activity against a range of bacterial species. Central to this section is the assertion that actinomycin D exhibits selective antibacterial activity, demonstrating potent effects against certain gram-positive bacteria, while displaying limited activity against gram-negative organisms. The authors present data, measured presumably using standard agar diffusion assays, to support this claim. It is important to evaluate this data critically. The zone of inhibition observed in such assays provides a relative measure of antibacterial activity. Factors such as the diffusion rate of the antibiotic through the agar, the growth rate of the test organism, and the inoculum density can all influence the size of the zone of inhibition. Therefore, a direct comparison of activity across different bacterial species requires careful consideration of these confounding variables. The observed selectivity raises important questions regarding the mechanism of action of actinomycin D. The differential susceptibility between gram-positive and gram-negative bacteria often reflects differences in cell wall structure and permeability barriers. Gram-negative bacteria possess an outer membrane that acts as a barrier to many antibiotics. The authors speculate on potential mechanisms, but the understanding of these mechanisms remained preliminary at this point. The claim that actinomycin D is particularly effective against certain species of Mycobacterium, specifically Mycobacterium tuberculosis, warrants particular attention. Tuberculosis constitutes a significant infectious disease burden, and the identification of an antibiotic with activity against this pathogen represents a notable advancement. However, further investigation is needed to assess the potential therapeutic efficacy of actinomycin D in treating tuberculosis. Section 4. Toxicity studies and in vivo activity. The section concerning toxicity studies and in vivo activity introduces claims regarding the toxicity profile of actinomycin D and its effectiveness in animal models of infection. The initial claim focuses on assessing the acute toxicity of actinomycin D in mice, a standard procedure for evaluating the safety of new drugs. The reported median lethal dose, or LD50, provides a quantitative measure of toxicity. In critically appraising this assessment, it is important to acknowledge the limitations of relying solely on acute toxicity data. Acute toxicity studies primarily serve to identify potential hazards associated with short-term exposure to the drug. They do not necessarily predict the long-term effects of chronic exposure. The authors also report on preliminary experiments evaluating the efficacy of actinomycin D in protecting mice infected with susceptible bacteria. This claim regarding in vivo protective effects needs careful examination. The experimental design, including the route of administration, the dosage regimen, and the timing of antibiotic administration relative to infection, all impact the outcome and interpretation of these experiments. It remains unclear whether the protective effects observed in mice would translate to similar efficacy in humans. Furthermore, the potential for the development of resistance during in vivo treatment adds another layer of complexity that requires examination. The observed toxicity raises concerns about its therapeutic potential. The authors acknowledge the need for further investigation to optimize the therapeutic index, which is the ratio of the effective dose to the toxic dose. A narrow therapeutic index implies that the drug must be administered with great care to avoid causing adverse effects. Section 5. Comparison with other antibiotics. This section presents claims comparing the properties of actinomycin D with those of other previously known antibiotics, striving to establish its unique position within the therapeutic landscape. The authors assert that actinomycin D exhibits a distinct antibacterial spectrum compared to existing antibiotics, suggesting its potential utility in treating infections, assistant to other agents. Evaluating such a claim requires a thorough comparison of the minimum inhibitory concentrations, or MIC, of actinomycin D and comparator antibiotics against a panel of bacterial strains. It is important to consider the potential for synergistic or antagonistic interactions between actinomycin D and other antibiotics. Combining antibiotics can sometimes enhance their efficacy or broaden a spectrum of activity. Alternatively, antagonistic interactions can diminish the effectiveness of one or both drugs. This section could benefit from a more comprehensive discussion of the potential advantages and disadvantages of actinomycin D compared to other antibiotics, weighing factors such as potency, spectrum of activity, toxicity, and cost. This comparative assessment is crucial for determining the true clinical utility of the newly discovered antibiotic. The authors suggest that the observed differences in antibacterial spectrum may reflect differences in the mechanism of action. While this is a plausible hypothesis, it requires further investigation to elucidate the molecular targets of actinomycin D and to compare them with the targets of other antibiotics. Section 6. Chemical properties and stability. The claims concerning the chemical properties and stability of actinomycin D provide insights into its physical characteristics and its behavior under different conditions. The authors assert that actinomycin D is a crystalline compound with limited solubility in water, but soluble in organic solvents. This claim regarding solubility has practical implications for formulation and administration of the drug. Poor water solubility can hinder the development of injectable formulations. The authors also report on the stability of actinomycin D at different temperatures and pH levels. This aspect of stability is critical for ensuring the potency of the drug during storage and handling. The authors show that actinomycin D retains its activity when stored in solution at low temperatures, but degrades at higher temperatures. This emphasizes the importance of proper storage conditions. Understanding the chemical properties and stability of actinomycin D requires a thorough investigation of its chemical structure. While the authors do not explicitly elucidate the complete chemical structure in this initial report, they do provide some clues based on its spectral properties. Further structural analysis would be necessary to fully characterize the compound and to understand the relationship between its structure and its biological activity. Section 7. Mechanism of action. Initial hypotheses. This section focuses on claims related to initial hypotheses concerning the mechanism of action of actinomycin D. The authors posit that actinomycin D exerts its antibacterial effects by interfering with essential cellular processes, potentially targeting DNA or RNA synthesis. This assertion, although speculative at this stage, represents a crucial step in understanding how the antibiotic works. The authors suggest that actinomycin D may bind to DNA, thereby inhibiting its replication or transcription. This hypothesis aligns with the observed activity against rapidly dividing cells and offers a potential explanation for its selective toxicity. Critically evaluating this initial hypothesis demands further investigation at the molecular level. Techniques such as DNA binding assays and studies on RNA polymerase activity could provide evidence to support or refute this claim. The authors also acknowledge the potential for actinomycin D to interact with other cellular components besides DNA and RNA, exploring alternative mechanisms such as interference with protein synthesis, is essential for gaining a complete understanding of its mode of action. Understanding the mechanism of action of actinomycin D is crucial for several reasons. First, it provides insights into the fundamental cellular processes that are essential for bacterial growth. Second, it can facilitate the development of more potent and selective analogues of the antibiotic. Finally, it can help to predict and prevent the emergence of resistance mechanisms. Section 8. Implications for chemotherapy. The final section of the 1952 paper addresses the implications of actinomycin D for chemotherapy, discussing its potential applications in treating bacterial infections and potentially other diseases. The authors assert that Actinomycin D represents a promising new antibiotic with the potential to combat infections caused by bacteria resistant to other agents. This claim must be tempered by the observed toxicity and the limited spectrum of activity. The authors cautiously suggest that Actinomycin D may also have applications in treating certain types of cancer. This line of speculation stems from the observation that Actinomycin D inhibits cell division, a characteristic shared by many chemotherapeutic agents. Later studies would reveal its significance in the treatment of certain childhood cancers. It is important to acknowledge the challenges associated with developing Actinomycin D as a chemotherapeutic agent. The high toxicity and the potential for the development of resistance pose significant hurdles. The authors underscore the need for further research to fully explore the therapeutic potential of Actinomycin D, to optimize its dosage regimen, and to identify strategies for minimizing its toxicity. Conclusion: Waksman and Schatz' initial report on Actinomycin D presents a detailed account of its discovery, production, antimicrobial properties, and initial toxicity assessments. The claims made within the paper are supported by experimental data, although the limited scope of the study necessitates cautious interpretation. While the presented evidence supported the potential of Actinomycin D as a novel antibiotic, it also revealed significant challenges regarding toxicity. This early work, though a foundation, would become the basis of future research. Subsequent investigations shed light on its mechanism of action, clarifying its role as an inhibitor of RNA transcription and revealing its unique therapeutic potential in treating certain cancers. The journey of Actinomycin D, from its initial discovery as a powerful antibacterial agent to its current application as a chemotherapeutic drug, exemplifies the complex and often unpredictable trajectory of scientific discovery. The initial claims, evaluated in the light of later developments, highlight the enduring significance of this 1952 publication as a seminal contribution to the fields of both antibiotic research and cancer therapy. The value, while not what the authors may have expected, is no less significant.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.